For today's episode, we will cover autism and Parkinson's. A major region of interest for both autism and Parkinson's is the basal ganglia. And sticking with this theme, we will cover some more basal ganglia, specifically the Substantia nigra. The Substantia nigra is in the midbrain, sometimes called the mesencephalon, sometimes called the tegmentum, and sometimes referred to as a region of the brainstem. Essentially, the Substantia nigra is towards the top of the brainstem. The translation of Substantia nigra is black substance or black matter. This is because of the neuromelanin. Remember neuromelanin's roles, and it is black. Because it is black, it receives all frequencies of light. Mother Nature uses it because it is black. It was selected for specific roles. Before I say the roles, this is true with the locus coeruleus as well. Same area and same black substance. Same cause for the black, neuromelanin. Remember the locus coeruleus for epinephrine and norepinephrine. You could call it brain adrenaline. And that internal calculator we covered with the astrocytes, a type of glia cell. This internal calculator that we covered was calculating effort versus outcome. And if the outcome does not match, the glia shuts the locus coeruleus off. So melanin, the synthesis of melanin comes from tyrosine, an aromatic amino acid. Those UV light detectors, 200 nanometer to 400 nanometer light. And tyrosine peaks at 280 nanometer. The strongest wavelength that arrives on terrestrial land from the sun. 280 nanometer light starts the UVB range, which is 280 nanometer to 315. Remember nanometer. There's 1,000 nanometers in 1 micron. 1 millimeter. And a sheet of paper is 80 to 90 microns, 80 to 90 millimeters. These are very small, and it fits the theme because of how small, how upstream we are going into our biology. The reason I say that is, this is as upstream as we can think about going. At least for humans and our ability to measure and see things. This is quantum biology, environmental signals. How living organisms on Earth are created. The sources of energy, how cells receive energy, which provides energy to the living organism. At the atomic level, how the powerhouses of the cells, remember the mitochondria, how the powerhouses of the cells receive the energy, the powers to power the powerhouses. Cells respond to environmental signals. Remember the 2 episodes on mitochondria, and remember the cause of autism episode. Water production is coming from Cytochrome Oxidase. And a decrease of this water production from the mitochondria equals less energy. This is huge for today's topic. Autism is underdeveloped cells. Parkinson's is that process running in reverse. Cells die because they lose energy. Melanin drives transduction, a transfer of energy from the environment into and through our biology. That's it. That's all you need to know about this. That's all you need to know about autism. If you can't go here, if you don't allow yourself to go here and allow yourself to understand this, you won't understand anything with our health and life. You will not understand autism. You might think that the research in autism is pretty lost. It seems like there's so much to cover. It seems like we're not getting anywhere. And this is true. Centralized autism research misses this in large part. It misses there is no power to the so-called powerhouse of the cell. And if you think about an effective fix, the treatment for Parkinson's is deep brain stimulation. An electrode placed in these areas to supplement the loss of energy. Energy lost from our modern environments that neglect the environment we evolved under. Humans do this, and no other species will do this. Okay, the roles of neuromelanin, this dark pigment. It's an antioxidant properties. Neuromelanin is our greatest defense to environmental toxins. It binds metals. This is a huge topic in current autism. It's well established. It is well recognized and accepted that environmental toxins cause autism or increase the risk of autism. But what's missing is what's changed. A top topic in autism research. It binds to atom and ions, and it makes it less reactive, less toxic, less harmful. Other ways of describing this are binding or sequestering. This allows detox of the cells. Thereby, it's a neuroprotection. A common and a region of interest for health is ROS, reactive oxygen species, or redox. Okay, so a lot here, and we cannot cover this. ROS is a very lengthy discussion, but ROS can be harmful to mitochondria and cells and proteins, lipids, and DNA. Next is free radicals. This is of interest. We've covered some free radicals when discussing blue light, tech light, and or isolated wavelengths of light. The primary region of interest here, the primary region of interest for modern health. Next for melanin is a neuronal regulation. Another easy connection, especially with dopamine. Also, electrons, the energy source from earlier. What powers the powerhouses? Remember, Cytochrome Oxidase. Reverse engineer the ATP process to the electron transport chain, to those cycles pumping out enzymes and ions and so forth, all the way back to pyruvate and TCA cycle, uric acid, which is a very hot topic in Parkinson's. Just reverse this final output of the mitochondria, the ATP, and just backtrack, and you can find implications here from the loss of environmental signals, from the loss of transduction. And remember chromophores, the red light chromophores on cytochrome oxidase, and the vitamin D receptors. More and more on this light topic. And humanity has seen rapid changes in light over the past 130 years. The question is, what do you think light is? Think about it. Are we using the way we evolved? Or have we changed light as a convenience? What do you think light is? And is it the same today versus evolution of life on Earth? This is so simple if you understand Dr. Jack Kruse's orange tree example. If you block the sun from an orange tree, will it produce the same amount of fruit? And even if the production of the fruit, and also for the oranges coming in being bloomed, will those be fully developed? Or maybe they're not fully developed. I use an example of this in the episode with Dr. Richard Fry when we discussed understanding the roles and sensitivity of mitochondrial functioning. And I use this example for describing autism and Alzheimer's. And there's a short on YouTube that you can find if you're interested in this. With neuromelanin, with Parkinson's, hypoxia of these pigmented neurons break down melanin. The source, the power, and the electrons. This creates a loss of dopamine. Now, it's common that people think Parkinson's is a loss of dopamine, and that's true. But research often misses the neuromelanin aspect of this component of this biological energy, these biological concepts. So let's go upstream in the biological path. The eyes, hair, and skin. This is the starting points, the starting lines. Think about this in a linear path. If the first domino doesn't hit the next, what happens? The eyes, we have melanin here. Of course we do, lots of melanin. We have melanopsin, and we have the retinal pigmental epithelium, or RPE. With this linear path, the goal here for this energy transduction is to remain efficient and powerful down the line, down the path. To answer the question earlier, what is light? Melanin is chosen here because it is a dark pigment and absorbs all frequency of light. In the summer, if you go outside and you wear black versus white, there's going to be a big difference, a noticeable difference. This is why the pupil is black. This is why Mother Nature chose neuromelanin for this role. Melanin and the pupil are black to receive the full light frequencies. So light is electromagnetic field containing a wave and a particle, a photon, a wave-particle duality. Waves have the information. Photons carry the energy. In the linear example of the photon, or photons, hitting the first domino, the eye, skin, or hair, it must be as much power as possible to power the downstream processes. Biologically, this is how we make the dopamine, serotonin, proteins, whatever the example like these you want to use. Vitamin D is a popular example. Most people understand sunlight and vitamin D. You can see the cause of autism episode for more detail on this, and also autism and gastrointestinal problems episode. There's a lot of this transduction happening in the gut. Artificial light versus sunlight is the cause. UV light wavelengths keep the melanin and dopamine optimized in the midbrain. The Substantia nigra for this Parkinson's discussion. Tyrosine is an aromatic amino acid. Remember those UV light wavelengths. Do you think artificial light is optimizing these biological processes? Don't miss the wavelengths throughout the light spectrum. This is why I ask, what do you think light is? This is also occurring on the skin with melanocytes. Something well known in autism research is thyroid and the correlation with the risk. Thyroid is tyrosine and iodine. Iodine is well established in autism as well. This is hitting us right in the face. Thyroid and Parkinson's is similar. There are strong data here. People with hypothyroidism have an increased risk of Parkinson's. Mothers with hypothyroidism, their offspring will have a higher risk of autism. This is all right here in front of us. We have all of these siloed data and research studies, but we cannot connect them because we don't go upstream. Because research has this centralized framework and is very confusing. Now, in our modern world, the thyroid is a pretty frequent lab test because the rates of thyroid are increasing. It's pretty common to find people, especially adults, with hypothyroidism. And we can think about the hypothalamus is the control center for the body's hormones, and then it communicates to the pituitary. And based off of these lab tests, you're probably even familiar in your own medical history. You know about T3 and T4. The 3 and the 4 represents iodine. So the T3 is tyrosine, thyroxine, and iodine. If you look up autism research, one of the ways to mitigate the risk of autism is to supplement iodine. Another way that we mitigate risk factors for autism are supplementing with things like thyroxine medication. And this is well established and it works. It mitigates the risk of autism when the mother is supplementing with these medications. So the important part here is the roles of T3, the roles of the thyroid with neuronal function and motor function. This is well established, but I don't think it's well understood or appreciated. So the roles of T3 here for brain development. This is during the fetal development. During fetal development and even in through early childhood, this T3 is critical for brain growth. It has a big influence on cell differentiation and migration. Remember the episode on autism and the embryo, and the episode covering the placenta. This is a key region of interest here. This is going to be when autism is developing in the living organism. So the T3 with the differentiation and migration, autism research has well-established data on these being a problem in autism. In addition, T3's role with maturation of neurons also another well-established data point in autism. Now, the overarching goal here with this process, building the brain development, is for cognitive functions. We think about autism, it's underdeveloped. It's a neurodevelopmental problem. T3 also helps with neuronal activity. So once the cell, the neuron, is formed, T3 regulates the gene expression in the neuron. And this affects neurotransmitter synthesis and receptor expression and synaptic plasticity. All of these things are well established in the autism data. T3 also acts as a neuroprotector, similar to the neuromelanin, and T3 has roles with reducing oxidative stress and aiding in repair processes after cell injury. T3 has roles with metabolism and energy, which is well established. The lay public likely knows this. However, it's the metabolic rate of the neurons, influencing the energy availability for the neuronal firing and maintaining of the neuronal health, cell health. These all have big implications with the synaptic transmission as well, and the gene expression. So if you're thinking T3 and the pituitary and the hypothyroidism, that's all associated with hormones. And that's correct. T3 acts as a connector, like a node here, that's connecting the hormones and all of these growth factors and the neuronal activity. Now, if you go downstream with these implications, it's easier for the lay public and just anybody to observe these. You might think of them as such as cognitive impairments or mood swings, some neurological symptoms even. One might go to the doctor and say, I just haven't been feeling well. My energy is low. I'm irritable. And they will do lab work. And well, indeed, it'll show your TSH might be 7 or 8 or 9 or whatever it is. And then they'll put you on, and you might be prescribed a couple of micrograms, 75, 100 micrograms of this medication. Now you can also understand the relationship here with the motor functioning. At the upstream level here, everything's related to cell health, the development of cells. If we lack energy, the cell lacks energy. So it's always coming back to the lack of or the loss of energy. For autism, this is happening in the womb, early in development, on this linear timeline. And we can move this out, stretch the timeline across these basal ganglia areas, and understand the problems here with the efficiency of this pathway. Something that's looking at us right in the face with this data are the environment of the pregnant mother has changed drastically. The rates of autism matches the type of environment the mothers are in. And it's not a direct shot at the mother. It's just the modern world, the modern environment. We are losing energy to the environment. Now, if you talk to a researcher on mitochondria, they might think, yeah, it's a mitochondrial dysfunction, but they might not know why. And that's not a shot against them either. It's just this centralized framework that determines which lens are we looking through. What are we looking at? What are we looking for? It's very siloed. It's very narrow. It almost lacks depth, if I had to sum it up. Now remember, thyroid role in the biosynthesis of tyrosine here as well. I cannot harp on this enough. Remember the biosynthesis of tyrosine, making thyroxine and making L-DOPA, which is common with the Parkinson's theme, and making dopamine and epinephrine and norepinephrine and melanin. Melanin is the missing role here. It's the underrated role. Remember melanin's role with water. So the mitochondria is here, once again. And the regions with this most neuromelanin are centered in the midbrain, areas that contains lots of other water in the brain. The melanin and water interaction equals more electrons. Remember, light changes the physics of water. This is what it is all about, electrons and the environmental signals. And remember the water being produced out of mitochondria. As we age, roughly each decade of life, we lose roughly 10% of the water being produced in mitochondria. And this occurs across the lifespan. 10% roughly every 10 years. So we are losing energy naturally. This is entropy. This is senescence. This is life. Aging and life. Check this with heteroplasmy. If you want a good marker, a good biological marker, look into heteroplasmy. Now, with disease and other insults and such, we can lose more frequency of water. This rate can be increased. Now you can even begin to understand the other neurodegenerative problems and why the rates of these are increasing. Okay, I said in a previous episode, because we're covering the basal ganglia quite extensively here, I would cover the direct pathway and the indirect pathway in the basal ganglia. So I'll do that now. Quick recap on the basal ganglia. There are 5 subcortical nuclei, all working together to orchestrate movements. The basal ganglia is our go, no-go areas. And this is where motivation and movements converge. But remember, the definition of motivation here is not defined by us and our ability to think and create with our human cortex. Motivation here is defined by the specific living organism. The nervous system just wants to respond. So that is the definition that you need to consider for motivation. The nervous system is just going to respond based off of what it knows. This is a very different definition of motivation. So remember the 5 subcortical areas. There are inputs, so the caudate nucleus and the putamen. And then we have the globus pallidus internal and globus pallidus external. These form a triangle, 2 separate nuclei forming a triangle. And we also have the subthalamic nucleus, which is the personal assistant essentially to the thalamus. And of course the substantia nigra, the 2 nuclei here, the reticulata and the compacta. So these are all orchestrating movements based off of the environmental signals and the perception of the living organism's nervous system. We are orchestrating movements here. The direct pathway is trying to facilitate movements by reducing inhibitory outputs from these basal ganglia nuclei into the thalamus. This will send a signal up to the cortex for responses deemed necessary or efficient or practical, useful for the living organism. The start is neurons from the cortex, all various areas of the cortex, and some from the thalamus and some from the substantia nigra providing dopamine. These inputs are not exhaustive, but just kind of to explain the pathway. The direct pathway acts as more of the go signal. So we're receiving excitatory dopamine from the substantia nigra. This is D1-like dopamine, excitatory. The dorsal striatum then will communicate with the globus pallidus internal and the substantia nigra reticulata. it communicates to these two subdivisions by inhibiting them. It puts a brake on them because the roles of the globus pallidus internal and the substantia nigra reticulata is normally to interact with the thalamus. So the dorsal striatum will shut these two nuclei off, and that frees up the thalamus. And the thalamus, being less inhibited, can then communicate back to the cortex, exciting the cortex and promoting movements. This pathway removes the brake, removes the no-go side. The indirect pathway is a little bit more communication. It inhibits or kind of slows down movements, acting as a brake. So one is a go side and one is a no-go side. Similar to the direct pathway, signals come in to the input area, the caudate and putamen, from the cortex and the thalamus and the midbrain. The dorsal striatum will then speak to the globus pallidus external, and it will inhibit the external area. By inhibiting the globus pallidus external, it will free up the subthalamic nucleus. It allows this area to be more active. Then the subthalamic nucleus will then speak to the globus pallidus internal and the substantia nigra reticulata, essentially giving them free will, giving them a go-ahead signal to activate. Remember, the direct pathway inhibited these. So here we are exciting these two areas. The globus pallidus internal and the substantia nigra reticulata then speaks to the thalamus, the main goal. And these two nuclei to the thalamus says, stop. This is a no-go. And because of the thalamus is inhibited or stopped, it can suppress or even slow down movements. It controls the movements. The indirect pathway is the no-go. And it uses the D2-like dopamine, the inhibitory dopamine. What the basal ganglia is trying to accomplish here is fine-tuning of motor control. Remember the goal of the central nervous system. It's designed to move the living organism. What these two pathways are doing is fine-tuning of that motor control, this action selection. And remember, the dorsal striatum is an area where goal-directed movements and behaviors are located, and also habit formation. Our habits reside here. There's a lot of communication from sensory motor regions and the dorsal lateral side of the striatum, the DLS, whereas the goal-directed resides more in the dorsal medial striatum, which is more caudate. So caudate is more go direction, and the putamen is more habit formation. It's where habits live. And that's the goal of the central nervous system, though. We want to build learning and then send those down into habits. And these areas of the basal ganglia and this direct pathway and indirect pathway, this is how we can grasp our motor signals being processed and leading to how we coordinate downstream behaviors, downstream movements. Now, Parkinson's here is a loss of timing and loss of energy. Now, with everything with the substantia nigra compacta and reticulata, you can start to understand why. The substantia nigra here, losing energy, losing its ability, its efficiency. It cannot conduct this pathway correctly. It loses its ability to coordinate these two, direct pathway and indirect pathway. And if you think about it, with all the with the motor movements associated with Parkinson's and autism. This is pretty common and easy to understand here. The Substantia nigra is lacking here, and all of these downstream processes are off. So with autism, we get these stereotyped motor movements and stimming and repetitive restricted behaviors and so forth. In both Parkinson's and autism, it's easy to see. We love it when we can see things and observe and evaluate things. It's easy to see the motor movements here, the abnormal motor movements. In autism, it's well established that there's an increase of this D2-like dopamine facilitated through the dorsal striatum and is implicated in the indirect pathway. We cannot inhibit motor movements. And over time, remember the discussions on neuroplasticity. These motor movements can actually become calming. We learn how to do these for calming, and they also remember the habit formation. We do these out of habit now. The more we do things, the easier it is to form the habit, which is the goal of the central nervous system, because it doesn't want to work. It just wants to respond. And considering that the Substantia nigra is not optimized, the transduction is not capable, its loss of energy is very implicated here. This is what it's all about. If you look at go versus no-go and put them on a seesaw, the fulcrum there, the thing that it's balancing on, is the Substantia nigra and the dopamine coming out of the Substantia nigra and into these other nuclei, facilitating these movements. If you are listening to the episode or listening to the podcast, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586, and we can discuss anything about autism. I very much appreciate your comments and interaction. You can check out the Hop link so you can have links to all of the shows across various podcasts. You can contact me on YouTube. Check out the YouTube channel. And TikTok. I guess I'll try out TikTok. I'll see how that goes. I don't love it. But anyways, thank you for listening to From the Spectrum podcast.